So today I am going to tell you about DNA, and most of you already know that a DNA looks like this. You've probably seen it in many textbooks, and you know that it is a unit of hereditary. So it is passed on from generations to generations, and also it it codes for everything. It stores information. It stores information on how to code for proteins, on like your hair color, your gender, and everything that you are made of is because of this gene. So now that you know that this gene is what makes you you, let's see what this gene itself, or the DNA actually, is made up of. So DNA. So we say deoxy. Oh, I'm so sorry, my pen is dried up. Deoxyribonucleic acid, right? So this is made up of three things, and they are the pentose sugar, a nitrogenous base, and a phosphate backbone, phosphate molecule. So you've seen the double helical structure, and there are two strands going. One is purple and the other one is pink. And the lines in between are actually what code for your proteins and everything. They're storing the information. So these two ribbon-like structures, the pink and the blue, are collectively known as the sugar phosphate backbone. And that is because they are made up of this pentose sugar and the phosphate molecule. So if you were to take this spiral ladder type molecule and actually just expand it a bit, you are gonna get what looks like almost a ladder. It is a ladder, and you have these steps which you climb on. So this is a ladder, and so you know that these two are the phosphate backbone, and this individual steps would be your nitrogenous base. And the reason why they're color coded and almost like half of each is because each one of these is a nitrogenous base. So this orange is a nitrogenous base, this green is a nitrogenous base, and this blue and the red are all nitrogenous bases. But first, let's talk about the pentose sugar. So a pentose sugar is a 5-carbon sugar molecule. And what that means is that it has 5 carbon in it. And it is a pentagon. You could draw a pentagon, and a pentagon would have 5 sides. So each edge would represent a carbon molecule. And so I've drawn this basic structure over here. And so over here you can see that it's a pentagon, but on the top there is a circle, and the circle is an oxygen. It is not a carbon. And now you might wonder, now how is there going to be 5? I thought it was a 5-carbon sugar. So over here there is 1 carbon, and then again here another carbon, here's another, and again on this edge there is a bond that goes from this carbon to another carbon, which is bonded to 2 hydrogens and 1 hydroxide over here. So this here is the 5th carbon. And so we like to number it, so we would call this 1, 2, 3, 4, and 5. And so our pentose sugar in the DNA is either ribose or deoxyribose. And in the DNA, your D stands for deoxyribo, which comes from the sugar molecule. And RNA has ribose in it, so it has this ribose. But what is the difference between ribose and deoxyribose? The only difference between ribose and deoxyribose is 1 less oxygen molecule. So a deoxyribose has one less oxygen molecule, and I would first draw you a ribose molecule to show you the position of the oxygen and the hydrogens and carbon over here, and then we would compare it with the deoxyribose, which has one less sugar. So take this backbone again, pentagon again. So here's a carbon, all these edges are carbons. We would put here a CH2OH because I had told you there's a fifth carbon there. And now on each of these lines that go on the edges, they are actually representing bonds, and on the bonds they are bonded with H and OH. Again, H and OH, H, OH, and on this end it is just going to be a H, and this here would be your ribose sugar. Now you want to know a deoxyribose structure, right? So you're going to draw another pentagon and draw those bonds, and you're going to say, okay, this is again your CH2OH, your H, OH, and on the second carbon, instead of this O over here, you're just going to have one H. Again a H, OH, and again an H. So now you see there is one less oxygen. So this here would be your ribose sugar. Next would be your phosphate group, and the phosphate group is essentially your PO4 2 minus. And how would you draw that? You would draw a P, which is phosphorus, and your oxygen. So it has 3 oxygens single bonded to it and 1 oxygen double bonded to it, and they have 2 negative charges, which you're going to denote there. So generally when we talk about the structure of DNA, we don't really consider the molecular structure. I mean, it's good to know what they are, but it's better, I mean, it's less complicating if you just draw it out like a pentagon or you know, another shape that could represent what you're drawing. And I'd like to represent phosphate as a triangle or a circle when I draw out a simpler and a bigger view of the DNA when we look at that later, how they bond together and why the DNA is the double helix that it is. So next we would go on to the nitrogenous bases. As I told you earlier, each one of these are the nitrogenous bases. So that gives us 4 nitrogenous bases. 4 nitrogenous bases. And all these 4 are actually divided into 2 groups. 2 groups. And what are those groups? Well, those groups are the purines and the pyrimidines. And this is where they differentiate purines and pyrimidines because of their structure. And where they do that, purines are molecules that have a double ring structure in them, as you can see over here. This molecule has a double ring structure and this molecule has a double ring structure as well, and they come under purines, and these 2 come under pyrimidines. And okay, so let's name them. This one over here is adenine or adenine. This one here is guanine. This one is thymine, and again this is cytosine. And you might be wondering what this tiny molecule is over here that I've drawn. This is uracil, and it is in RNA only. It's RNA only, and it replaces thymine in it. So next time, I mean, when you have an RNA strand and you have an adenine, it would bond with the uracil, not the thymine. And we'd come back to that bonding later. It's generally speaking, it is A, T, and G, and C. Adenine bonds with thymine, guanine with cytosine, but I would show you how that works later on. But for now, you could just know that always adenine would bond with thymine and guanine would always bond with cytosine to give you this piece of the ladder. So you have 2 parts of it, and one is A and one is T, or one is G and C. It's never gonna be A and C or T and G and stuff like that. It's always adenine to thymine, guanine to cytosine.